Hello and welcome back to our first tissue mini lecture from chapter 4. This is going to be on epithelial tissues. And just to kind of heads up, epithelial tissues and connective tissues are the largest of the two categories. Muscular and nervous are pretty short. So if these first couple of videos are going to seem really long, I apologize. It's just, I'd rather just do a few semi-long ones instead of trying to break up epithelial and connective. So just bear with me for the epithelial and connective tissues. You'll get a break in the length of the videos when we get to muscle and nervous. All right, so let's just jump right in. So epithelial tissues, before we get into the specific types, just some unique characteristics that we might see associated with epithelial tissues. So this is just a generalized what we would call a simple columnar epithelium, but I'm going to use it as a diagram to illustrate some of these unique characteristics that we see in epithelial tissues that we may not see in other tissue types. So I'm just going to move from the top down on this diagram. So when we are talking about cilia, cilia are extensions of the cell that are usually used for propulsion. They act like little beating hairs that we have them lining our respiratory tract, in our trachea and in our upper respiratory tract. They help to propel mucus and things like that. So cilia are for propulsion, coordinated beating action to move things along. Whereas microvilli are also little extensions off of the surface of the plasma membrane, but instead of functioning for propulsion, their job is to increase surface area. So for example, if we had a cell that looked like this versus a cell that looked like this, if I were to be able to draw better, their volumes are about the same. But if we were to measure the surface area from here compared to the surface area here, we'd have a lot more surface area on the cell with a microvilli than we would just flat. So make sure you connect microvilli with absorption, increasing surface area for the function of absorption, and cilia for propulsion. There we go. The next term here we have what's called the apical surface compared to the basolateral surfaces. So those terms kind of go together. Apical means towards the exposed edge, the exposed surface. Basolateral is the attachment point. So basal is on the basement membrane, lateral could be to its neighbor. So by definition, epithelial tissues are tissues that cover surfaces, they line surfaces and open spaces. So you're always going to have an attached edge and you're always going to have a free edge. The attached edge is called the basal surface, and the free edge is called the apical surface. So just adding new vocabulary to our understanding. So instead of calling it base and free, we're going to call it basal and apical. So just keep that in mind. And then they're just adding lateral to kind of go up here. So basal, lateral surfaces, apical surfaces. So if we wanted to use that in a sentence to put it in context, we could say cilia and microvilli are located on the apical surfaces of epithelial cells, where the basolateral or the basement membrane is connected, is found at the basolateral surfaces, or the nucleus is closer to the basal surface and the cilia are found at the apical surface, or the lumen of the digestive tract is in contact with the apical surface of those digestive cells. So just keep that in mind, the usage of those terms. And then the other thing are just your organelles, right? So Golgi, nucleus, mitochondria, these other things. It's just, basic cell biology. Typically in a column-shaped cell like this, most of your organelles will be closer to the basal surface, but sometimes you have a square cell like this, and everybody's just kind of crammed around. There's not a lot of free space. Okay, and then lastly, the last term we need to cover is the basement membrane. The basement membrane is a connective tissue membrane, and it's like proteins and some extracellular fibers, and that is what the epithelial cells are connected to. So we're going to see that here in another slide where we actually talk about connections, but there's little protein anchors that help to hold the actual plasma membrane of the cell onto this basement membrane. So all epithelial tissues, because again, the definition is they're lining things, they have to be attached to something. And that thing that they're attached to is the basement membrane, okay? So those are some structural characteristics. I think the notes also say things like regenerative. So epithelial tissues are regenerative. They have the ability to repair themselves and replace themselves if damaged. And epithelial tissues are cellular. It's like, well, duh, aren't all tissues cellular? Well, yes, but remember on that in the intro video, we talked about extracellular matrix. Epithelial tissues do not contain an extracellular matrix. They're just all cells. There's nothing extra outside of the cell components of the epithelial tissues, okay? So here is some of the ways that these cells, since it's all cells, how they're connected to each other. So there's quite a few different intercellular connections. We have gap junctions, tight junctions, and desmosomes. We have what are called spot desmosomes and hemidesmosomes. Okay, so those are the 4 connections we're going to talk about on this slide. So gap junctions are connections between neighboring cells that allow things to freely move in either direction, usually following osmosis or diffusion. So things go from high to low concentration. But basically what it allows for is neighboring cells to share ions or nutrients or things like that. So gap junctions are, like they imply, gaps. So they have holes that are open between the neighboring cells. In tight junctions are usually towards the apical surface, and what they do is they basically seal the neighboring plasma membranes so tight that not even water can go between them. So the reason why, and I would normally have drawn this on the whiteboard in class, so bear with me here. So if we had some cells, here's our basement membrane, say in the digestive tract. And so we have nutrients and stuff, kind of these nutrients and things up here, or maybe it's bacteria, or just things in the lumen of the digestive tract. Cattail. And we don't want them to seep in between the neighboring cells to get down into the underlying tissues. That would be bad in that particular location. So what these tight junctions do is they come along and they make these tight seals. And it prevents these items from being able to seep in between the cell. So tight junctions are found in locations where you only want the substances to be regulated through the cell, not in between the cell, okay? And then desmosomes and hemidesmosomes, or spot and hemi. Desmosomes are really strong anchors. So they are the things that are really anchoring the cells to the basement membrane and anchoring the cells to each other to make a really strong continuous covering over a blood vessel or skin or a digestive lining. So spot desmosomes are going to be between the cells, and you can see there's these little anchoring proteins and they're hooked to the cytoskeleton, where in a hemidesmosome, hemi means half, right? So we've got half of this little protein circle kind of dense area, and it's looping in. So here's a plasma membrane, that's a phospholipid bilayer, and then here's our basement membrane made up of clear and dense layers of connective tissue, and then we find these little anchoring proteins that loop around and anchor the cell cytoskeleton to those underlying connective tissues of the basement membrane. All right, so those are intercellular connections. Gap junctions allow for communication between the cell. Tight junctions prevent leaking of things, I guess you could say, in between neighboring cells. And think of hemidesmosomes and spot desmosomes as strong structural anchors holding the cells to each other and holding the cells to the basement membrane. Okay, so now we're going to get to starting to see some examples of different kinds of epithelia, talking about where they can be found and what they do. But first, we have to talk about how they are classified, how they are named. So again, epithelial tissue, because they're covering surfaces, they're always going to have a basement membrane and a free edge, right? That was our basilar surface and our apical surface. Basilar and apical, always. That's the definition of an epithelial tissue. So if you can find your basilar surface, so each one, basilar surface, apical surface, basilar surface, apical surface, basilar surface, apical surface, right? And then once you find those boundaries, look at the shape of the cell. So the shape is either squamous, because it's flat. Squamous means flat, like a pancake or a fried egg. Cuboidal, as you can see, the cells are kind of cube-shaped. And columnar, they're tall and column-shaped. So those are the shape names. Okay? Squamous is flat, cuboidal is square, cube-shaped, columnar is like a column. Then we want to take a look and find those boundaries, right? So basilar surface, apical surface, basilar surface, apical, basilar, apical. And then we can count if there's more than one layer. If there's just one layer between the basilar and apical surface, it's called simple. So all of these three, single, squamous, single, cuboidal, and columnar, there's just one layer. So whenever there's one layer, we just call it simple. And then whatever the shape is. So if it's one layer of flattened cells, it's called simple squamous. If it's one layer of cuboidal cells, simple cuboidal. If it's one layer of columnar cells, simple columnar. Pretty easy, right? If you see from basilar surface to apical surface, there's more than one, two or more, then it's called stratified. It's a very bad square or rectangle. So here we look at the shape of the cell at the topmost layer, because that could be a little bit confusing because you're like, Corey, that looks cuboidal and that looks squamous. How am I supposed to know? Is it stratified cuboidal or is it stratified squamous? Well, the clue is you look at the uppermost layer, and we'll see this in some examples here in a second. So if we look at the apical layers, those are flattened cells. So this is going to be a stratified squamous epithelium. This one's pretty easy because they're all kind of cube-shaped. So this is stratified cuboidal, which is pretty rare. We don't even see this in our labs this week. And then again, it looks like there's some cuboidal cells down here, but if we look at the uppermost layer, this is going to be stratified columnar. And again, those are pretty rare, so we don't see them very often. So if you can start getting comfortable identifying the boundaries, right, basement membrane to free edge, you look at the shape, you look at the number of layers, and that's going to give you the name of these tissues. There's a few exceptions we'll get to, but this is kind of the big naming scheme for epithelial tissue. So simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, stratified columnar epithelium. And we have two exceptions to that. All right, my computer kind of paused out for me, so hopefully there's no skipping of information. All right, so we went through the classification, kind of the naming of most of the epithelial tissues. So now I'm going to show you some microscope slides of the different groups. So stratified, cuboidal, and columnar, with our two exceptions of transitional and pseudostratified. So on this slide, I am showing both simple and stratified squamous. So again, squamous, we're looking for the flattened cells. So maybe, let's see, maybe orange will be a good color so it won't get blended in with anything. So the top two boxes are examples of simple squamous epithelium. So I'm going to let you take a look at them, pause the video if you wanted to. I know they're labeled in this one here, so that's not cheating, but you don't get an opportunity to look. So for this picture here, see if you can find where the simple squamous epithelium would be. It's a little bit tricky, especially with some new histology eyes that you have, but we're looking for a basement membrane and a free edge with a flattened, single layer of flattened cells between the basement membrane and the free edge. Now, if I could try to put my brain back in the beginning when I was first learning tissues, I'd be like, yeah, there's no way I can find that. So if you're feeling that way, that's okay. I'm going to show you how. So here is the basement membrane and all the way around, and here is the free edge. So that tiny little layer of cells from the basement membrane here to the free edge here. It's the lining of this structure in the kidney called the glomerulus. All right, so here's basement membrane. Here's free edge. And you can kind of see some nuclei, right? So there's a nucleus, There's a few nuclei that got caught in that slice. So that would be an example of simple squamous. Now over here, they're labeled for you, but I will highlight, here's where the basement membrane is going to be. Now you can't really clearly see the basement membrane. You can kind of see a little bit of a delineation. And here's going to be the free edge. So if we kind of identify those boundaries, here's one cell, like right here. It's one simple squamous cell. And then this is a lining of a capillary. This is in the lungs. So then in the alveoli, which is the air sac, here's the basement membrane, and here's the free edge, right? So single layers of flattened shaped cells, simple squamous. Now, functionally, simple squamous are really good for diffusing things because it's the thinnest layer of epithelium that you have. So for our lungs, it's really good for the exchange of gases. So oxygen goes this way, carbon dioxide goes this way, and all they have to do is diffuse across that two thin cells and a little bit of this loose connective tissue. Okay, so functionally, you're going to see simple squamous epithelium in places where you have lots of diffusion or exchange surfaces. Now, the bottom two pictures are showing stratified squamous epithelium. So take a look at those, pause the video if you want to, see if you can identify the basement membrane, the basilar surface, and the free edge of where the stratified squamous epithelium is going to be. Okay, pause it or take a second. So in this purple one, I'm going to use my orange and I'm going to outline. This is where the basilar surface is, and you can kind of tell a difference just taking a look at the texture between the kind of the lighter pink and then the darker purple. And then here's my free edge. So between the basement membrane and the free edge, there's clearly more than one layer of cell, right? And then this pink one over here, so there's my basement, and there's my free edge up here. Many layers. Now let's look for the shape. So we want the uppermost layers. So you can kind of see these cells up here are kind of flattened like a pancake. They're not all of them are super clear. This one's a little bit easier. Look at this kind of bracket right here. Those are nice flattened shaped cells. So this is going to be stratified squamous. And stratified squamous functionally are going to be found in places where there needs to be protection. It's thick, it's usually keratinized, which means it's going to have this waterproofing protein in it. They're strong. The upper layers of cells are dead. So our epidermis, this is epidermis that I've just highlighted in both these bottom pictures, made up of stratified squamous epithelium. All right, so let's move on to another type of epithelium. All right, so this is going to be our simple and stratified cuboidal epithelium. So again, I'll give you a moment. You can either pause the video or I will just take a moment and first, again, when you're trying to identify epithelial cells, find the basement membrane, find the free edge, and see the shape and count the layers. So that should be your kind of habit when you're looking for epithelial cells. Okay, so I'm going to start in this upper left picture. So this is simple cuboidal epithelium. This is found in your kidney tubules. Now this looks a little bit weird, right? So you might be saying, well, that looks like it's stratified, but there's two free edges. So that shouldn't make sense to you. So let's erase one of those lines. And where would the basement membrane be? Well, it's this line right here. So that's a basement membrane on that side. And then here's the basement membrane on this side and the free edge. So we're looking at two tubes kind of stacked like this. And each tube is lined by simple cuboidal epithelium. So here are cube-shaped cells, just like that, simple cuboidal. And so here'd be another one up here, basement membrane, free edge, and then this is the lumen. This is where the urine is going to be traveling through as your kidney makes pee, right? Lined by these simple cuboidal epithelium. Now let's go down to this one. This is also simple cuboidal epithelium, but it also has that kind of weird back-to-back look to it. So the basement membrane is actually going to be right here, kind of surrounding these, what are called follicles. This is from your thyroid gland, right? So things like this. So those are all the basement membranes. So then here's the free edges that are lining these hollow spaces within these storage tanks inside your thyroid gland. These are the cells that make your thyroid hormone, And the thyroid hormone like T3, T4 are stored in those follicles. And then sometimes it's really hard to see the definition of the plasma membrane, but you can see the nuclei. So each one of those nuclei, we are assuming, is going to be within a single cell. So if we kind of divide those nuclei, then we can kind of see, oh yeah, they are kind of cube shaped. So there's your simple cuboidal. Now up here in this upper right-hand picture, this I'm showing an example of stratified cuboidal epithelium. Again, it's a little bit more challenging. We don't have a slide of stratified cuboidal in the lab, but so I'll give you kind of some starter. So here's the basement membrane. Right. So now you got to find yourself a free edge. Tiny little lumen right here. So not much to that. So between the basement membrane and the free edge, right, if we're going to put cubes around each of the nuclei, we almost have 2 layers of cube-shaped cells. Okay? So stratified cuboidal, we're not going to see as many layers as like we would see in stratified squamous, but we see enough to identify it as stratified cuboidal. Okay, so functionally, we have simple cuboidal lines things, again, for diffusion and osmosis, so good for exchange of things. Stratified cuboidal, very rarely found in the ducts of some glands, and it's primarily for protection. It doesn't really do much absorption because it's thicker. You're not going to do much absorption like diffusion or osmosis through things that are stratified. Simple, yes. Stratified, no. And that's true for both squamous and cuboidal. All right, so move on to our columnar. So again, kind of this little histology quiz. Can you identify the basement membrane? Can you identify the free edge? Can you identify the shape and what's going on here? So pause the video if you want to try it on your own, or we'll just keep on going. So the first one in the upper left, kind of the lamina propria, that's our basement membrane, so we can identify that here. Here's our free edge, right, because there's nothing, this is all open space out here. And then again, this one you can kind of see the boundaries of the plasma membranes. So column-shaped cells, makes sense. And then here's the other basement membrane, and these are kind of pointing down that way. So that's pretty straightforward. And then here, the basement membrane kind of goes up. You can see that difference between the purpley connective tissue underneath and then the free edge here. So it's kind of going around like a big loop. This is in your intestines, like that. So there's my basement membrane, there's my free edge, and then I can see these tall, skinny cells like this. That's my simple columnar. All right, now down here we have our stratified. So again, you can kind of see that subtle difference in organization where the basement membrane is going to be. And then here's the free edge. On both of these, again, we're kind of lining a tube. So here's the lumen. Right? So now this is a little bit tricky. These stratifieds are not super easy. So here we have a little layer of what almost looks to be like cuboidal cells, then with these taller, skinnier cells on top. Right? So again, when we're trying to identify stratified shape, you look at the most apical layer. So this is a stratified columnar, even though it looks like that innermost layer is a cuboidal. So for function, and location, I guess. Most simple columnars found lining your digestive tract, and its function is for absorption. A lot of times these are going to have microvilli. We can't see those in the microscope. Microvilli are just too small. But the increased surface area allows for increased absorption. So simple columnars primarily absorption. Stratified columnar, again, rarely found in the lining of some ducts of some glands, also for protection. Excuse me. All right, so those are three kind of that combination of simple and stratified, squamous, cuboidal, and columnar. Let's take a look at some exceptions. So these are the exceptions. We have transitional epithelium and pseudostratified ciliated columnar epithelium. That's a mouthful. So let's take a look at the stratified first. It's the two right-hand images, the ones that are kind of above my head here. So again, pause the video if you wanted to try to decipher these yourselves, try to find the basement membrane and the free edge, or we'll follow along here. So here's the basement membrane, Again, you're looking for that delineation of texture is your biggest clue. And then here is your free edge. Over here, here's the basement membrane. And here's the free edge. Now, upon first appearance, you're like, well, that looks like stratified cuboidal, right? So here we have many layers and we have these cube-shaped cells at the top. So how come this isn't stratified cuboidal? Well, a couple of reasons. One is it doesn't always look like this. It's called transitional epithelium because it changes its shape depending on what's going on in the body. Transitional epithelium is primarily found lining your urinary bladder. Right? Urinary bladder starts out about the size of an acorn, and then it can fill up to about the size of a grapefruit, and then you pee it out, and it goes like this, back to the size of a walnut. Did I say acorn? Walnut? They're about the same size, right? And then it goes to about this, and then you pee. However many times you fill up your bladder and pee every day, your bladder is going like this, and it's lined by this transitional epithelium. So when your bladder is empty, it's big and puffy like this. When your bladder is full, that transitional epithelium actually stretches so flat that it almost looks like a simple squamous epithelium. So we typically only see it in the bunched up, kind of the empty bladder form. And so this is our transitional epithelium, and you can recognize it because of the dome shaped. I tried to accentuate that in my little drawings here. So these are dome shaped cells because they're all kind of scrunched and pushed up on themselves. So transitional, it looks like stratified cuboidal, but it's not. Those dome-shaped cells. And true stratified cuboidal usually only goes to 2 layers thick. This is way too many layers to be a true stratified cuboidal. Okay, found in the lining of the urinary bladder and the ureters, I believe. And it also, for functionality, it just allows for the stretching and recoil of those organs. The next one is our pseudostratified ciliated columnar epithelium. So pseudo, what does pseudo mean? Pseudo means false, right? Stratified means layered. So if something is pseudostratified, it should look stratified, but is not truly stratified. And then cilia means it's got cilia, and columnar means it's column-shaped cells. So again, let's try to find, pause if you want to, the basement membrane and the free edge. So here we go. Basement membrane, free edge. Basement membrane, free edge. Okay, so now we've identified basement membrane and free edge. We can look at the shape. They definitely look columnar. They kind of look like they could be layered, right? So here's some cells, or it looks like maybe some column-shaped cells. So query, how come it's not stratified cuboidal or stratified columnar? Well, remember, stratified columnar is pretty much just 2 layers, kind of like our stratified cuboidal, and stratified columnar is never ciliated. So the cilia are good clues. And then also seeing that some of these cells kind of have this teardrop shape or kind of this point to them. A lot of them have that, like we can see that down here. So that's also a clue you're looking at pseudostratified. So even though it looks stratified because the multiple layers of the nuclei, each cell is attached to the basement membrane. There's not cells attached to cells attached to cells. So they are all attached to the basement membrane. Some of them make it to the surface, some of them don't, but there's no layering of the cells. And then you also have the cilia, is a good clue. This pseudostratified ciliated columnar epithelium is found lining the trachea, some of your upper respiratory passageways, and it's also found, ciliated tissue is found in the reproductive tracts, like the epididymis in the male, the uterine tubes in the female, helps to propel, right? So all of these little cilia beat in unison, and the mucus or the egg or the sperm gets propelled along through that tube or space, okay? So that is our transitional and pseudostratified ciliated columnar epithelium. And let's see, where are we at? Okay, I think what I'm gonna do, no, let's just keep on going. I know we're at a half an hour, but I don't wanna do a whole other video for glands. So we'll just keep on trucking here in epithelial tissues. So those are all of our different types of epithelium, but epithelia are also our modified glands. So our 2 major groups of glands are exocrine, get back into the red, exocrine and endocrine. Exocrine are called such because they secrete their product onto a surface. It could be the surface, like your skin, but it could also be the surface of, say, like your oral cavity, like salivary glands, or in your stomach, your acid and your mucus going into the inner surface of your stomach, or your digestive tract or your respiratory tract. So a surface, not just the surface of the skin. So a surface. And then endocrine sends their secretions, called hormones, into the blood. So I'm trying to write with my left hand. Blood. It's horrible writing on my screen. I'm gonna try again. So endocrine sends their secretions into the blood. That's better. So up here, I have a picture of actually what's called a mixed gland. The pancreas has both endocrine and exocrine. So you can see the blue cells are secreting into the pancreatic duct, which will eventually lead to the surface of the duodenum, where the endocrine, this is where your glucagon and your insulin goes directly into the bloodstream. They don't have a duct. Sometimes they're called ductless glands. Excuse me, I'm not used to talking so much this summer. All right, so those are different types of secretions, exocrine versus endocrine. And now we have different mechanisms of how those secretions, the exocrine, well, we have a whole chapter of endocrine we're going to cover in 232. So we're going to focus primarily on the exocrine for the rest of this chapter and this discussion, I mean. So depending on where these exocrine secreting cells are located and what they're secreting, they'll have different mechanisms of secretion. So there's three major types. We have what's called merocrine, apocrine, and holocrine. And again, these are only dealing with exocrine secretions going to a surface. In the first one, called merocrine, the example of this is going to be one of your salivary glands. Is called your parotid salivary gland. They're showing in that drawing here. The secretion is saliva. And the cells do just normal exocytosis. So if you remember learning about that back in your general bio, the cells make the product, transcription, translation, making these proteins. They get packaged by the Golgi or endoplasmic reticulum in the Golgi body. The vesicles move to the apical surface, and then they exocytose and just release just the product into the duct, and then that will go into your oral cavity. That's your saliva. For apocrine mechanism of secretion, the example of this would be the mammary gland for breast milk. The production starts the same, so it's produced, transcription, translation, the protein products modified by the endoplasmic reticulum and the Golgi, packaged into the vesicles and kind of headed towards the apical surface. But instead of exocytosing, the top half of the cell starts to pinch off, and it pinches off and breaks away, and the whole top half of the cell is what gets released into the duct and eventually out through the lactiferous ducts and sinuses for breastfeeding. So it's not only just the breast milk, but it's also a little bit of cytoplasm. It's also a little bit of plasma membrane becomes the breast milk product. And then the cell will repair itself because they have that ability to do that. In holocrine secretion, the example here is your sebaceous glands. So that's like your oil glands on your hair and your face and your skin. So again, everything's kind of started the same way. The production of the secretion, in this case, it's called sebum, S-E-B-U-M, packages up in the little vesicles, but instead of exocytosing or just pinching off the top half, the whole cell breaks open and releases all the components, the secretion, the nucleus, the cytoplasm, all the organelles go into the duct. But because of that, you have to have some stem cells located at the very basilar surface. It's usually a stratified structure. And so the top cells, these all die. But you can see some cells going through mitosis to replace the cells that are lost as they continue to make this secretion. All right, so that's called holocrine. So merocrine, apocrine, and holocrine mechanism secretion for exocrine secretions, not endocrine, not hormones. Things like, saliva, oil, sweat, mammary glands, earwax, mucus in your nose, all of those would be secreted by these exocrine secretions by varying mechanisms, the merocrine, apocrine, or holocrine. Okay, we're doing last slide. So then this slide is just showing you different structures of how these exocrine glands can be formed. They're just just variations on what we're going to be seeing in some of our slides, like when we get to the integumentary system and other slides. So we have two groups. We have simple and compound. So simple just means you have one duct. I'll put it in purple because that's kind of their duct color. So the duct is purple, so there's one duct leading to the surface, one set of duct cells, right? And then I don't have pink, but we can just do red. And then we have the secretory cells. So the name of the gland is a combination of how many ducts to the surface they have, and then what's the shape of the secretory cells. So you have simple, all the top row is simple because it's one duct to the surface. We have tube-shaped secretory cells, coiled tubes, branched tubes, alveolar, which means like a sphere, and branched alveolar. So we got tubes and branches, or sorry, tubes and spheres. And then sometimes they're coiled, sometimes they're branched. And then for compound glands, the reason why it's called compound glands is because there's more than one collection of duct cells. Now, there really is just one duct to the surface, right? We can see that there's just one duct. However, if we look close enough, there's other collections of duct cells, right? Those are not secretory cells. They don't secrete, they just structure. Okay, so wherever I put the purple, those are duct cells. And since there's more than just one set of those, that makes them compound. And then we can take a look at their shape. So it can be compound tubes, compound circles called acinar or alveolar, and compound tubes and acinar. Tubuloalveolar is what they're called. So this is just giving you some names to the shapes of how these exocrine glands can be found in your tissues. All right, that wraps it up. I know, 36 minutes. That's probably going to be the longest. Connective tissue is also going to be a little bit on the long side. But remember, I said muscle and nervous are going to be on the shorter side, so that'll be good. All right, I will see you next time. There's my other kitty. Right there, I got two kitties. They'll be walking around behind me this term as I'm recording my videos. Okay, so see you next time for connective tissues. Bye.